Hello, everyone, and welcome. I'm Susan Hedstrom, and I will be your moderator on today's webinar. We're fortunate to have several newly funded studies to share with you today, as well as updates on our progress as we work to develop treatments for PWS. But before we get started, let's review a few housekeeping items. You are on mute. However, we would love to hear from you, so please submit any questions you have through your GoToWebinar control panel, and we will answer as many of these questions as we can at the end of the presentation. This presentation is being recorded and it will be made available after the webinar so that you can review any slides or content after today's presentation. If you have not already done so, please consider signing up for our blog and newsletter so that you receive updates on our research programs, as well as the link to the recorded webinar. And without further ado, I'd like to give you over to Dr. Theresa Strong, who'll be sharing details today on our funded projects.
Great, thank you so much, Susan, and thanks everybody for joining us today. I know it's a very busy time of year, and so I really appreciate you taking a little bit of time to join us and learn about some of the new projects that we're going to be funding in 2021. So I'm Theresa Strong. If you don't know me already, this is my son, Daniel, who has PWS. He's 26, and this is Rex, his dog. That's them hiking about. And so, you know, getting a little bit of fresh air as we all have been doing during the pandemic. So today's agenda, I'm going to try to keep it pretty brief. You know, I get excited about talking about all this research, but I will try to control myself a bit. I'm going to give you an update on some of the PWS research activities that are going on, some of the returns on investment, you know, things that came out this year that maybe you missed, maybe you didn't, and then talk a little bit about the new grants that we funded in 2020, and happy to have a discussion at the end, as Susan mentioned. I always say thank you at the end, but I wanted to make sure that I said thank you at the beginning because it has been such a crazy, extraordinary year, and extraordinary not in a great way in many aspects. I did want to thank our development team at FPWR, Jackie Maizan, Melissa Demand, Sarah Peden, you know, for being there and thinking creatively and helping us maintain our fundraising throughout this pandemic. I mean, there have been so many other demands on all of us, and they have thought really creatively. And I especially wanted to thank all of our donors, whether, you know, you fundraised on Facebook, or you had a virtual event, or you bid on one of our art auctions. You know, we really appreciate your ability to focus on PWS research in this time that was really, again, very challenging for everybody. I know everybody was dealing with a lot. So because of you, we are able to keep this research moving forward. The scientists have continued to work, although there's been a few stoppages here and there along the way, but we've actually made some amazing progress given all that's going on. So thank you at the beginning. And some of that is shown here. I mean, when I step back and think about where we were 5 years ago versus today with respect to treatments that are in the pipeline that are being tested in individuals with PWS, it is really remarkable. I mean, 5 years ago, there was maybe one clinical trial going on. We've completed more than 15 clinical trials with 9 different potential treatments with PWS. And are really making progress towards those new treatments. Pharmaceutical companies have invested in the PWS community to get these projects done. If I think about the studies that have been done to date, it's more than $250 million of pharmaceutical companies investing in new treatments for PWS. So that is, again, truly remarkable. Now, we don't have a new approved treatment yet, but you know, you have to have shots on goals in order to move forward. And as we know, we got 3 phase 3 clinical trials done in 2020: Levo's carbetocin, Soleno's diazoxide, and Melendo's levolitide. Levolitide did not show efficacy, and that program is halted, but the diazoxide and the carbetocin showed some promising results, and those companies are talking to the FDA about what are the next steps to move towards approval. In addition, we're talking to additional companies, and we're supporting additional more basic research that is leading to new drugs that might be applicable to PWS, and we will continue to push those forward. So it's an exciting time, and you know, hopefully 2020 is going to push us into a brighter 2021 where we can get some new therapies out to our kids. So I'll encourage you to pay attention to the clinical trials page. Of course, clinical trials were slowed by the pandemic, but they're starting to pick up again. Pitolisant, Harmony Biosciences is currently recruiting for their Pitolisant clinical trial. We'll talk about a study using guanfacine for regression, and Eric Hollander's group has a FPWR-supported study in CBDV in the New York area that is currently recruiting. So sign up for the clinical trial update, you know, visit the website often, and you can see where the new clinical sites are. We will be seeing additional clinical trials likely coming online in 2021, so please stay in touch about those. And I'd also encourage you to continue to update your records in the Global PWS Registry. That has been such a useful tool to understand the natural history of PWS, to support these clinical studies. Many of the pharmaceutical companies have asked us for information about the population based on the information that you have put in. We're sharing that de-identified, of course, with both the scientists, the clinicians, and the pharmaceutical companies to support research, support improved care in PWS, and support new clinical trials. And in 2021 January, we're going to have a little push to update your surveys. Some of you have been in the registry for a long time. You've got a lot going on, so it's hard to remember to go in and update your records. So we're going to try to have a nice push in January to get all of those records updated, because we are getting a lot of requests from PWS clinicians to pull data, and we want to make sure it's the most up-to-date, it's the most representative data that we can provide to them so that they can understand PWS. Okay, so today we're going to talk about the grant program that FPWR funds, and the grant program funds innovative research and exceptional opportunities across this entire research spectrum. So this is the research spectrum that we are keen to support, moving everywhere from understanding PWS and discovering new targets, all the way through the preclinical development, which is testing it in cells and in animal models, and into the clinical development where we test it in individuals with PWS. And our ultimate goal is to have new treatments available. So we have a lot of programs to support steps along the way, but the grant program, you know, we look for those exceptional opportunities anywhere along the spectrum that can help speed things up. So let's talk a minute about the grant program accomplishments. Since 2003, when FPWR came into existence, we've invested more than $14 million into PWS research, supporting over 175 research projects around the world. And that has led to over almost 200 publications now. And publications are great, but it's not just publications for publications reason. It's publications that advance our understanding of PWS and give us new options for treatment. In addition, FPWR-supported work has led to additional investments, as I mentioned, from the pharmaceutical industry, which is not included in this $25 million, but that $25 million includes investments from you know, other granting agencies like the NIH, for example, and new resources that can be used by many individuals in the PWS research community, as well as new drug targets and new candidate drugs. So here's some examples of papers that have come out in 2020 that were supported by FPWR, so supported by your funds. A couple of papers on diet from both Ann Scheinman, looking at the modified Atkins diet and how that works in PWS, as well as Andrea Hack, looking at, again, a low carb versus a higher carb diet. There have been some clinical trials and studies. Dr. Hollander's group has recently published the results of their oxytocin clinical trial, which showed, which actually did not show efficacy in their trial. So it adds to the literature. We now have oxytocin is very plus minus. There are several studies that show some benefit, particularly in selected subgroups, and there are other studies that are not showing benefit. So understanding why some people seem to benefit from oxytocin but others don't, I think is really going to be very important for our community. We also have research coming out from Dr. Anastasia Dimitropoulos' group showing how an intervention helping kids learn to play and interact socially can be important to their development. I think that's really important for our younger kids. And there have been some really important advancements in our understanding of the underlying defects in PWS. And there's one paper in particular, this paper by Dr. Potts and his group, that I think is really important. And I'm not going to go into it today because I recorded like a separate short version of what that's about. It really is defining why there's a decreased level of neuropeptides in individuals with PWS. So that will be coming out on our blog and on our website. So hopefully you can look at that. And I would encourage you to, just check our website often. We try to update all the time when new outcomes come out. We had additional outcomes in the area of genetics, understanding other genes in the PWS region, looking at genetic therapies and advancing the genetic therapies for PWS. So all of that is on our website. And of course, I'm happy to answer any questions about that. So let's talk about the new grants that we funded in fall of 2020. It is a little bit less grants than we had hoped to, because it has been a challenging year financially. Again, thank you to everyone who was able to donate, but we understand that it was a year of uncertainty, and so we tried to be a little bit more conservative this year as well. But I think we found some fantastic projects to support, and I'll tell you about those now. So the first is one you may actually recognize. I'll just bring it back up because it's starting to recruit now, and I think that is, or will be starting to recruit very soon. That is Dr. Singh, who is looking at guanfacine for aggression and self-injury in PWS, and he's doing a placebo-controlled clinical trial. And he was at NYU, and he has now moved. So the grant has been reissued to him in his new institution, which is Maimonides, a medical center in Brooklyn. So the rationale here is that aggressive behavior, of course, we know is very common in PWS, and it can have tremendous implications for the person with PWS as well as for their family. And Dr. Singh has had a few cases in which the drug guanfacine, which is an approved drug, has seemed to improve aggressive behavior in individuals with PWS. So this funding will allow him to do a controlled trial and really show that. And I think that's really important for our community to be able to show it in a controlled setting, to give everyone confidence that this is an approach that should be used, and it should be used or not, to help us determine that. So we're excited about this study because aggression and lack of impulse control, which underlies perhaps some of those behaviors, can be really significant issues in PWS, and they can really impede our children's ability to be successful in school and successful in work. Guanfacine is an approved drug with a well-characterized safety profile, so it has that advantage of knowing the drug, and it's been prescribed widely in typical individuals. But now it will be looked at specifically in PWS. We know also that out in the community, individuals with PWS are being prescribed some ADHD medications. This is typically used as an ADHD drug, but it's important to understand in our population what the efficacy is and what if there are any unexpected side effects. So that's what this study will help us do. It's a single site study in Brooklyn, New York, actually really close to where I was born, but that was a long time ago. Anyway, in our next group of grants, neurobiology, we're looking at energy balance, feeding, and cognition. So these are obviously really important areas for our kids, and we have 3 grants in this group. 2 that are looking at feeding behaviors and what underlies the circuitry of that, and 1 that's looking at cognition. So the first is from Dr. Joe at Albert Einstein, and there's a particular set of neurons, the POMC neurons in the brain, and they regulate energy intake, energy expenditure, glucose metabolism, all of which obviously are important in PWS. Previous research has shown that these POMC neurons don't function adequately in PWS mouse models and likely in PWS as well. So in this particular study, Dr. Joe is going to use mice that are lacking the MAGEL2 gene, which is one of the PWS genes, and look at it specifically in the POMC neurons, excuse me, to identify how that loss of MAGEL2 in those particular neurons impacts weight and obesity. So why are we interested in this? Well, Dr. Joe is an expert in the neurobiology of feeding behavior. He's made a series of important discoveries about how these POMC neurons function. And so now he's going to apply that expertise to PWS. The researchers are going to also look at how the POMC neurons impact a part of the brain, the amygdala, which is important in controlling motivation to eat, and food seeking, which we know is important in PWS. So these studies may provide a novel mouse model that mimics the energy balance disruptions that we see in PWS. So we know PWS is about, that desire to overeat, but it's also about that lack of energy balance. So, calories in really seem to stick on a person with PWS. So the long-term goal is to understand the cellular mechanisms underlying food intake and weight gain in PWS, and it may lead to improved treatments for overweight and obesity. A second study is looking at the neurohormonal controls of energy balance in a new rodent model of PWS. FPWR has supported the development of a MAGEL2 deficient rat model, and the rats allow us to look at more detail compared to the mice in some aspects. And so Dr. Metliki Bass is at SUNY Buffalo, and she is an expert in looking at feeding behaviors in rodents, including rats. So the rationale here is that the causes of energy balance, again in PWS, is not fully understood, and we can maybe use this new rat model to better understand that. So she'll be investigating the neural and hormonal controls of energy balance in this PWS rat. So why are we excited about this study? Well, we know that that disruption of energy balance, the kind of packing on the calories that our kids can do so easily over the lifetime, that makes a huge difference for individuals with PWS. It's so hard for them to lose weight and maintain a good, healthy weight. This is a relatively new animal model, and so we'll get to see, what new things it can tell us about PWS. And it's an in-depth exploration of feeding motivation, satiation, how satisfied we get from a meal, appetite hormones, and neural control of energy balance. And another nice thing about this is a young investigator who's new to PWS, but she has a lot of experience in how the brain integrates signals from the body to control food intake and body weight. So this will be a great addition to our PWS researchers. So again, the ultimate goal is to understand the physiology and neurobiology of energy balance in this new model of PWS. And the third grant in the neurobiology area is from Dr. Tim Wells, looking at the neuronal architecture in a mouse model for PWS. And this study is really focused on cognition. We know all individuals with PWS, to a greater or lesser degree, have some cognitive impairment. And Dr. Wells will be looking at, what is the basis for that? So we gave him a very small grant over the summer, and he and his students made some excellent progress looking at the architecture of the neurons, that's these down here, in PWS mice, and found some differences compared to typically developing mice. So they're going to now sort of expand that study, use a specialized cellular analysis that they have expertise in to characterize the architecture of a particular set of neurons that's really important in cognition and development. So we're excited about this study because it's a very experienced group of researchers, and it's going to address a major gap in our understanding of PWS. So there's a lot of work going on in other neurodevelopmental disorders to understand the basis of intellectual disability. And in PWS, there's really not that much going on. And I think that's going to be important because in some other intellectual disabilities, there are actually treatments that are going into clinical trials. So if we could have an understanding of how PWS compares to some of those other disorders, it might open up new options for improving cognition in our children and potentially adults with PWS. So these studies are going to extend the Wells Group's preliminary findings and help determine if there's an issue within the neuron itself. So they found some abnormalities. Is it actually the neuron, or is it the environment around the neuron? And that will help guide how you might think about intervening into that process. So in the long term, their goal is to identify the mechanisms of cognitive impairment in PWS and to begin to evaluate therapeutic strategies that might improve cognition in PWS. So we'll move on now to two studies that are looking at the cellular phenotypes of PWS. So I talked earlier about Dr. Potts doing some really exciting work, understanding what's different in a PWS cell compared to a normal cell. Those of you who have been, watching the PWS research world for a few years will know that we've made lots of different kinds of stem cells that have PWS, and so these two grants are really gonna rigorously look at those stem cells and kind of pull all the information we can out of them. How is a PWS neuron or brain cell different than a typical neuron, and how can we use that information to help guide us as we look for new therapies? So there's two groups that are going to be using a different set of approaches to the same question. I think it's important to have more than one group working on this because there are many different ways you can approach this question. And, as with all of our investigators, we will be working to help them collaborate so that they're sharing data so that we're gaining, even more information from the two groups together. So the first is from Dr. Tai, who is at Massachusetts General Hospital, the Center for Genomic Medicine, and they have an amazing sort of machine up there looking at several neurodevelopmental disorders. So this study is going to be, it's unraveling mechanisms of PWS by molecular dissection of driver genes in a hypothalamic neuron model. So the question here is, we know there are several genes in the PWS region, so how exactly does each of those contribute to the characteristics or phenotype that we see? And working in the Talkowski lab, so Dr. Tai has recently been promoted to a junior faculty member. He has been working in the Talkowski lab over the last couple of years on PWS, and he has helped develop this amazing panel of stem cells that, using CRISPR engineering, they've cut sort of big pieces and little pieces and single genes and regulatory sequences out from the PWS region. So they have a stem cell line that has the large deletion, and a stem cell line that has the small deletion, and the stem cell lines that has a tiny deletions. And by comparing all of those, they can really get a sense of what is specific to PWS, what are the differences between large and small deletion in PWS, for example. So they have this panel, and now in this study, they're going to characterize the cellular properties of these stem cells once they are differentiated or encouraged to become like hypothalamic neurons in a dish. So they take the stem cells, they throw some things on them, and they turn into these neurons that mimic hypothalamic neurons, which of course are really important in PWS. So they will apply cutting edge omics technology, proteomics, transcriptomics, all of the omics, which is like globally looking at the proteins and genes that are expressed in the cell to characterize how the PWS neurons are different than typical neurons. They'll integrate that information into the lab. So as I mentioned, the lab has an extensive pipeline looking at other neurodevelopmental disorders. So they have a lot of data to compare PWS to, and that's a real advantage. So they can see sort of what's shared and what's not shared, what's unique to PWS. So this overall is a powerful resource for the PWS community. These cells already are being widely used in the PWS community. The work by Dr. Potts's lab has included these cells as well. In the long term, the goal is to provide new cellular models for PWS, first to understand the changes in PWS, and secondly to identify new targets and new drugs that might help or restore some of those deficits as they're found. Another study looking at the cellular phenotypes is from Anne Bang, who is at Sanford Burnham Institute, and she will be developing a suite of assays for the analysis of PWS patient iPS-derived cortical neurons. So rather than using the CRISPR neurons that the other group is using, she's going to use patient-derived neurons that were developed by Stormy Chamberlain's lab at University of Connecticut, and she is going to apply assays. Dr. Bang is very experienced in high-throughput assays for neurons, so she's going to take that expertise and apply it to PWS. So they'll use, and this is the first step towards screening for drugs, right? Screening for new drugs that might treat PWS. So important throughout the brain. So we're excited about this study because it brings an experienced cell biologist who's at a premier drug screening institution who kind of does this, all the time to look at our PWS cells and really, apply their knowledge to that. The studies are going to provide a basis for understanding cognitive and psychiatric challenges in PWS. That's what cortical neurons are really important in, that thinking and mental function. And the goal is to develop rigorous cellular assays, which will then lay the groundwork for doing drug screens to see if there are ways, if we do find changes in the cortical neurons, are there drugs that can kind of encourage them to be more typical in their functioning. So the goal is to develop a validated PWS assays to be used in drug discovery efforts and eventually to actually do that screening. So our final project for this round is from Jim Resnick at University of Florida, and Jim has looked for a long time at the PWS region, understands the genetics really well, and is looking at this problem of genetic therapy for PWS. Can we turn the maternal genes on? If you'll recall, all individuals with PWS, whether they have deletion or whether they have imprinting and UPD, have the PWS genes sitting there. They're just turned off. So in thinking about genetic therapy, we have some basic questions like, when do we need to turn those on? And you can hear Dr. Resnick talk about some of those questions and about this project in the fireside chat from last year's meeting. But this project, it's going into year 2. And it's addressing, a couple of key questions. If we're going to do genetic therapy, when would we have to do it? Would we have to do it, before a child is born? Would it work in an adult? Would it have to be during the infancy? Those are really important questions as we move forward towards doing gene therapy. And so in year one, Dr. Resnick developed a mouse model that allows the genes to be turned on precisely at different times and in different cells. So that's been established. And now in year two, he's going to be using this mouse model to turn the genes on and see what the consequences are to really get at those questions. So this is an experienced investigator who has a deep understanding of the region. This is a new mouse model that will help scientists understand when gene therapy would have to take place, in what cells gene therapy would have to take place, if it's going to have an impact on individuals with PWS. Of course, this is in the mouse for now, but the idea is to set the stage for human studies. So this will establish the feasibility of genetic therapy for PWS, including the expected effects if you turn the genes on at different points during development. So those are the projects that we're going to be funding as we look forward to 2021. We have a lot of hope for certainly a better year than 2020 was. You know, some projects across the spectrum, because we fund projects across the states and across the world, there were some slowdowns, but by and large, the scientists kept plugging away at it, wearing masks now, being socially distanced, going into the lab at, at nighttime, so there's not too many people in at one point. Hopefully we'll get back to a more normal schedule where they can continue the research without quite such sort of hardship in the working environment. We're looking forward to new treatments for PWS. There'll be next steps in these interactions of the companies with the FDA, trying to move towards approval. We'll have start of several new clinical studies looking at different drugs that address things like sleep and appetite in PWS. We have some new insights into the biology of PWS and the molecular mechanisms, which sets the stage for thinking differently about what new treatments might affect lots of aspects of PWS at once. And that's, stemming mainly out of the work by Dr. Potts and all the colleagues that he worked with there. We're expecting, there's a newly established PWS clinical investigation collaborative, a network of PWS clinicians that are working together to address some of the clinical care issues in our loved ones with PWS. So we'll be looking for exciting work to come out of there, as well as the global registry. And our organization will continue to interact with the FDA, continue to take the patient perspective and convey it to pharmaceutical companies and to the FDA, and explain the unmet medical need and the needs of our community. So I believe that's all I have aside from the thank you. Please stay up to date. You can always email me or Jessica if you have questions about the grant program in particular, but me with any questions. And, all of the website, we try to keep that up to date. Carolyn Vranadias on our research team has been really busy trying to make sure that the most accurate and up-to-date information is there. So Susan, I'm happy to take questions.
Thank you so much, Theresa. It's always wonderful to hear you describe some of these projects. I know reading them in their descriptions can be, or even the titles, can be very intimidating. So having you come on board and really telling us what these projects mean is so important. We do have questions, of course, that are coming into the queue. I would encourage anyone who has a question to go ahead and type it into the question control panel, and we'll run through as many of these questions as we can. Theresa, they're coming in somewhat in backwards order. So I have some questions from the last slide that you just shared with us. I think this is the first one that's on most people's minds is, do we have an idea of when these new drugs, being carbetocin and DCCR, might get approvals?
So I knew that was going to be a question, and I would say that I'm not in a position to speak for the companies. They really need to express that directly to the community. I do know that both companies are discussing with the FDA what would be the next steps. Is the information that they have now adequate to move forward? Is there additional information needed? And they are I can I do know that they're working very hard on that. They're aware of the fact that the community is very anxious and, you know, really wants this to move forward. They're all over it. But those discussions back and forth between them and the FDA, you know, they just have to play out. So so I can't, not that I have any, you know, secret information, but even, you know, I just can't speak for them. They will communicate that directly. I wouldn't want to misstate anything. So so, you know, the drug approval process, once a new drug application is in, takes, I believe, 9 to 12 months. So that's a minimum.
Now, both of these drugs have fast track designation. Will that speed up that 9 to 12 month period?
I think, okay, so someone can correct me, but, and I should have looked at this before I think the 9 months is with the fast track designation, because I think it usually takes, you know, 18 months is a more typical amount of time.
All right. So let's answer a few questions in regards to genetic therapy. Will genetic therapy work for individuals who are UPD, who have the 2 copies of mom's genes and nothing from dad?
Yes, presumably it would, right? So the PWS genes, again, whether you have deletion, you've got 1 copy of them. If you have UPD or imprinting, you've got 2 copies of them. They are there. They're just not active. So much of the genetic therapy is to try to turn them on. And it may be different in UPD compared to deletion, right? So in UPD, you've got 2 copies. So is it going to be different if you turn both of those copies on as compared to deletion, where you just have 1 copy? That's a possibility. And so we'll need to look at that, right? We'll need to look at how it works in deletion versus how it works in UPD. But the basic concept, the genes are there. And, you know, now, at least in a dish, there's been several publications. One of them, if I go all the way back really quick, from Dr. Chamberlain's lab, using targeting a specific protein that regulates that on-off. And she has shown that, you know, you can turn the genes back on, and you can do that in deletion or in UPD.
So that leads nicely into the next question, which was, have there been any genetic therapy outcomes from some of the research that we funded that shows promise that this will work in Prader-Willi syndrome?
Well, a lot of those projects are ongoing. So we funded, so 2020, the early 2020 was, you know, a place where we put a lot of funds into genetic therapy. So I think we funded 4 or 5 projects that are using different approaches. So using a CRISPR-based approach and using, you know, an oligo-based approach and using, you know, a different kind of molecule to regulate the region. And so the idea is now, you know, just to give all of those options a possibility of playing out. So those are all ongoing. Those were all grants that were funded in the last spring. And so they're in the midst of getting done. So we would expect to see those coming out. You know, like what works best, I would expect in 2021, and the ones that look most promising then will be taking forward. But in the meantime, we have Dr. Resnick's work. Dr. Resnick is not really looking for a technique to turn those genes on. He's using a genetic trick in the mouse. But his project will answer, when do we need to do that? So on the one hand, we've got, okay, we're developing the techniques so that we can move it forward. On the other hand, we're asking the questions, how would you do it? When would you do it? You know, those kinds of things. So we can know when both of those things should come together to impact PWS.
It can be challenging for many of us to look at the project titles and know which ones are focused on genetic therapy. Can you give us an idea of how much of FPWR's programs are focused on advancing genetic therapies for PWS?
I can, it's off the top of my head, so it could be a little bit off, but I think in 2020, we, you know, in total, we have spent about $1.6 million towards grants this year, and about I would say 250. I think it's either 230 or 260. I can't remember, but about 250 of that was for genetic therapies. So we really made a push this year to support. And in the past, I'd have to go back and look at that question, because we have been supporting genetic therapies all along. Some of the, Dr. Young-Hui Zhang's work, that showed that initial proof of concept that you could turn the genes back on in a mouse model. And that was a couple of years ago. So we've been supporting projects all along. But in the last year, I would say probably about a fifth of our entire research budget was going towards genetic therapies. And I'd always like to see that increase. I mean, I think there's a lot of opportunity to coordinate among those groups and really push things forward. So I'm hopeful as we sort of get out of the financial constraints of the COVID era, that we can really put even more focus on that, because I think it's like the time is right to have all those people coordinate and decide on the best approaches and really move those forward.
Right. Do we know if the mRNA technology used to develop the COVID vaccines can provide any benefit for PWS treatments in the future?
So that's an interesting question. Potentially yes. I think the experience that's being gained with mRNA therapeutics in the COVID space will be, important for PWS. I could envision, just off the top of my head, using mRNA, a CRISPR-based mRNA, for example, to go in and edit the PWS region and sort of restore gene expression. I think, that's potentially a way to go. Gene replacement by mRNA is difficult because the mRNA is kind of, it's like invisible ink, right? So, and that's why it works so well for vaccines, right? You get the vaccine, it gives your cell the instructions to make the COVID or coronavirus spike protein, and then it disappears. So it doesn't integrate into your DNA, it doesn't mess with your DNA, it's just sort of there short term. So, so using it like as a gene replacement would be challenging in PWS, where you have to, you have to give genes for the entire lifespan of an individual. But using it to do gene editing, I think is really a powerful way to go. And there's a lot of work being done on that. And I would like to see, again, in a larger genetic therapy program, bringing that technology into PWS and using it to edit the genetics and potentially have a long-term impact. I don't know if that made sense, but the short answer is yes, there's a lot of potential application there.
Great. So you talked about a couple of projects that were using cells to perhaps develop new drugs for PWS. Can you describe for us a little bit the process? How are cells used to identify new drugs?
Yes, so for example, and I wish I had the pictures. So in this paper that the Potts lab published, what they showed is that these secretory granules there's less of them in PWS. And you can see them in the cell, they can stain them, so they're like little green dots. And what you see is, and if you watch the video that I recorded earlier, which I don't have right here, what you can see is in the cells, PWS cells, there's many less green dots, right? So what you can do is you can take a plate that has thousands of wells with those cells in it. You can throw thousands of drugs on the cells, and then you can ask in an automated manner, and they do this with robots, right? What are the drugs that cause there to be more green dots? So what are the drugs that cause there to be more secretory granules, bringing it back up to normal? And that's how these things are used. So this study suggests that it is a decrease in secretory granules that causes a decrease in the neuropeptides, and the neuropeptides are like oxytocin, vasopressin, growth hormone, all these things that we know is low in our kids. It seems like it's because these secretory granules are not forming. Now, if we can take take cells, we can see those secretory granules. If we can screen thousands of drugs, we may find a drug that is used for, I don't know what, like malaria treatment, that actually increases secretory granules. So that's the beauty of these screens, is they're unbiased screens. So you just take the cells, you just ask the question, which drug causes, there to be more secretory granules, and then you look at that drug and see if it makes sense in PWS. And the beauty of those screenings is there are libraries with thousands of compounds that are already FDA approved or already have been in humans. So they have, that's called repurposing, right? So they're used for one thing, but maybe they would apply here. So that's the kind of thing we're talking about. We're talking about, we already have one cellular phenotype, there may be others. And then if we can automate that, then we can screen thousands of drugs and maybe find something, and perhaps something that's already approved, that we could try in PWS.
Now, has this process ever been used before to find compounds that might be able to treat PWS?
Not PWS, but it's been used successfully to, find the new drugs that are, essentially curing people with cystic fibrosis, for example. That's really was the first in the field of rare disease, was the drugs that have recently come out in cystic fibrosis, where, it was all high throughput screening like this.
Mm-hmm. Well, wonderful. Thank you so much, Theresa, for joining us today and sharing this information about our funded projects. For all of our community members, each of these projects are described on our website should you want more information, and we will be putting clips from this video on each of those descriptions so that you can rewatch this if you would care to have any more information. Thank you everyone for joining us. Thank you for supporting us. We truly appreciate it. Have a wonderful holidays and Happy New Year.
Thanks, everybody.